Yep.
So I'll tell you a story, you know, maybe it's true, maybe it's not, about how Caleb came to study the things that he's going to tell us today. So it all happened in the summer of 2016, and we were in the Idea Factory. That was July, and we were trying to figure out all those, you know, splicing variations that you see here in the image. And you can see Caleb here, a young man. That was 2016. He was still young. With Anu trying to figure out what's going on and figuring out this mess. And during this, trying to figure out what's going on, and you can see a lot of complexity in those splicing variations that Caleb is going to talk about today, right? Basically what happened in the Idea Factory, which of course is named because, you know, that's where all the good ideas come from. Caleb was saying, all these splicing variants, you know, what a mess. How can I find what is actually functional here? And Anu, who was standing beside him as a senior PhD student in the lab, said, exactly, I think you should focus on solving this for your PhD, right? Now, maybe that happened, and maybe it didn't. But besides the joke, and just, you know, as any good lie, of course, there's a bit of truth here. We were there in July 2016. It is actually a place, a real place. It's called the Idea Factory. And we were actually posing for pictures to show how cool the Idea Factory is. That's the truth. But I found this image kind of telling because it tells a story about the path, Caleb's path, towards his PhD and many others, I guess, in many ways. Because what happened is, you know, when Caleb came and joined both our labs, me and Kristen, he was actually asking different questions. He was interested in, you know, cell differentiations for TH cells and, you know, really, and the functional consequences of this. And he spent a lot of time on this. And he did get a first author paper out of this, which I'm not even sure he's going to mention today, although it's a nice paper and it's really good work that he did. But the thing is, these challenges that he ran into during his PhD sort of led, as you'll see in his presentation, I hope, led to the questions that drove a lot of the research that he did. So he was interested in the functional consequences of functional splicing, but then the challenges with the data as it becomes big and all that and complex drove a lot of his major contributions to what is currently being doing in the lab. And he, I feel, really found his path later on in those projects and making really important contributions to the things that we do every day in the lab, in both labs actually, and multiple projects. And, you know, just to sort of, so that's, you know, it's a nice story about, you know, where you start going in your PhD is many times not what you end up doing, but, you know, depends on your talent and perseverance and finding your way, you find really good things to study and work and do great work. And that's a story of this as an illustration. So yeah, without further ado, I guess we can switch to Caleb to tell us about that.
Thank you, Yosef, for the kind introduction. Share my screen here. You guys can see it? Okay. So yeah, thank you, Yosef, for that introduction. There's definitely some grains of truth to that story. I agree. I think that's a good anecdote. And thank you, everyone, for joining me today for my thesis defense. Yeah, so the title of my dissertation is Characterizing Local Splicing Variations from Heterogeneous RNA-Seq Datasets. But the most biologically relevant part of this title is just one word, splicing. And I'm going to talk a lot today about the biological relevance of splicing. So first, I kind of want to give an analogy, excuse me, of for splicing. I like to think of splicing as kind of like our cells having a copy editor to remove nonsensical punctuation. So take, for example, the classic example of the panda eats shoots and leaves. A good copy editor probably knows what you meant by the sentence. You probably meant the panda eats shoots and leaves, like the bamboo leaves. But if those commas were not removed, which is kind of what happens when splicing is disrupted, your sentence has quite a different meaning. Now it reads as the panda eats, then it shoots, and then it leaves. And now we have two completely different sentences, just because we've spliced out the commas or not. But this is actually where biology gets really interesting, because we can also alternatively splice out words to create entirely new sentences. For example, here I've spliced out the word eats. So now the panda just shoots and leaves. So this is my cute example of splicing, or analogy for splicing. But to connect this back to real biology, you can kind of think of DNA as having genes, where the genes are transcribed into precursor RNA, which are like the sentences before copy editing. Genes usually comprise a series of exons and introns, where the exons in boxes are like the words, and the introns as lines are like the nonsensical commas that need to be spliced out. After splicing, you're just left with exons and a fully spliced messenger RNA. And the messenger RNA is then translated into protein. Alternative splicing, however, results in distinct isoforms of messenger RNAs with differential inclusion of exons. For example, the red exon is only included in the bottom isoform here. Thus, through alternative splicing, one gene can encode multiple distinct protein products. And this process of splicing is carried out by the spliceosome, which is the machinery in our cells that recognizes specific sequences in RNA, such as 5' splice sites, branch sites, and 3' splice sites, to catalyze the splicing together of exons and removal of introns. Alternative splicing, however, is controlled by splicing factors. And these splicing factors either promote or inhibit recruitment of the spliceosome components, like U2, U2AF1, or U2AF, or U1. For example, splicing factors can act as activators, recruiting the spliceosome, or splicing factors can act as repressors, inhibiting recruitment of the spliceosome. So it may not come as a surprise that mutations in genes important for splicing can cause disease. For example, spinal muscular atrophy is caused by mutations in a gene called SMN, which encodes an RNA binding protein important for splicing. But what's really cool is there's actually a highly specific therapy to correct splicing in patients with spinal muscular atrophy. So there's good motivation to study alternative splicing in disease contexts. And RNA-Seq is an experimental method that facilitates research of disease-associated missplicing by enabling researchers to study splicing in samples of RNA from patients or RNA from mice or cell lines with specific splicing factor mutations to model a disease. And so this experimental technique, RNA-Seq, at a high level, you can think of the input RNA to RNA-Seq as a book full of sentences making up words. And the goal is to identify and quantify the sentences in the book. And so the RNA-Seq method first shreds up the book and then makes thousands of copies of the shredded pieces of paper. And then it's up to researchers like myself to try and piece back together the original words and sentences that were in the book. And if this seems like a messy and complicated way to study splicing, that's because it is. And as it turns out, there are lots of different approaches to identify and quantify these quote-unquote sentences, by which I really mean messenger RNAs. The approach that I've used in my PhD is as follows. I quantify splicing at local splicing variations, or LSVs, from the point of view of which, for example, from exon 2 in this gene cartoon, allows us to quantify the percent of RNA-Seq-derived junction-spanning reads going from exon 2 to 3 in red, 2 to 4 in blue, or 2 to 5 in green. And since 4 out of 10 of the reads here in this LSV splice to exon 3, the junction that's red is 40% included. or we like to say it has 40% psi. The tool that I use to do this is Magique, which was developed in the Barrage lab. And early on in my PhD, I was primarily using Magique to study splicing in simple models of disease. But as I progressed, I encountered increasingly complicated population-level and tissue-specific human data, and as a consequence have grown as a computational scientist over time, with the overarching PhD goal to develop and apply pipelines and tools to dissect biological signals from complex splicing data. And early on in my PhD, this meant discovering functionally relevant RNA isoforms, and later on, this meant developing methods to overcome technical challenges associated with heterogeneous RNA-Seq data. So today, rather than talk about all of my dissertation work, I'm going to focus on 3 stories representative of my dissertation. The first story used a mouse model to identify alternatively spliced genes important for inner ear development. The story started with an observation in the clinic that a pair of patients with hearing loss harbored loss-of-function mutations in the splicing factor ESRP1. But at the time, it wasn't clear what role ESRP1 plays in the ear. So my role in the project was to use RNA-Seq of mice knocked out for ESRP1 to identify genes whose splicing depends on ESRP1. Then to characterize how these genes were misspliced and prioritize which genes for my collaborator, Alex here, to follow up. This work is published, and feel free to download it and take a look. So my analysis of the RNA-Seq from these mice identified 518 ESRP1-dependent LSVs. And as a part of this project, I developed a series of Python scripts to analyze the junction coordinates of these 518 LSVs and determine how exactly splicing was disrupted in the target genes. So, for example, I found that 154 of these LSVs corresponded to simple exon inclusion or skipping. Most of the other LSVs also corresponded to simple binary events, meaning there was a binary switch in isoform usage between the control and knockout mice. For example, I identified in FGFR2 a pair of mutually exclusive exons. This splice graph here on the right shows how in control samples, the major isoform includes the blue exon 12 and skips the red exon 13. Whereas in ESRP1 knockout mice, the major isoform includes exon 13 in red and skips exon 12. And as it turns out, the ESRP1 dependent inclusion of exon 12 results in an isoform encoding a cellular receptor critical for development of the inner ear. So the patients with ESRP1 mutations end up producing the wrong isoform in the inner ear during embryonic development, leading to hearing loss. Another interesting example from my analysis is of a multi-exon skipping cassette in the gene COL11A2. I'm showing here at the bottom, and it appears pretty complicated, and actually exon 7 in green was not previously known to exist. But despite how complicated this appears, I determined that this event behaves in a binary-like fashion because there are only 2 major isoforms in the control and knockout mice. But this actually raised a fundamental problem that Joseph was alluding to in my introduction, and that's how do you determine what the major isoforms are? Why did I count COL11A2 as a binary-like simple multi-exon cassette and not as one of the 119 complex events that involved 3 or more isoforms? And my solution in this project was to focus on the most relevant junctions, which made it much clearer what the 2 main isoforms are. COL11A2, for the most part, either skips multiple exons or includes these 3. So for the ESRP1 project, I developed a series of Python scripts to accomplish this task, but I wasn't fully satisfied with my solution. I still had 119 out of the 500 events I was only able to label as complex. So this actually motivated me to develop a classifier algorithm that would be able to handle complex events. And I've been leading the development of this algorithm but a software developer in Yosef's lab, Paul Jewell, has been helping me to implement it. There are three main steps to the classifier algorithm. First step of the algorithm is to focus on the biologically relevant junctions. We call this step decomplexifying. And it's in Urban Dictionary now, actually, you can look it up. What I mean is, if you focus on the red lines, which are spliced junctions, and the associated read numbers, on average, you can see hundreds to thousands of reads per junction. The decomplexifying stage filters away junctions that don't have much evidence in your data of existing, and thus are probably not particularly relevant to your sample. So for example, here I've decomplexified junctions with less than 200 reads. And now the picture of which splicing features are alternative becomes much clearer after decomplexifying the splice graph. The next step is to identify alternative splicing modules, or modules, which are non-overlapping splits in a splice graph. And since I have a splice graph, I treat exons as nodes and junctions as edges, connecting those nodes in a directed manner. And then we can easily find non-overlapping splits in the graph as having a single entry and a single exit. For example, there are two modules in this splice graph. And the last step of the classifier algorithm is to identify the events within the modules. In this case, we have simple binary cassette exon skipping events. So as a proof of concept, I applied my classifier algorithm to one of the most heterogeneous datasets I could find, and that was 2,335 brain samples from GTEx. More specifically, I was interested in identifying genes that differentially splice between 13 brain subregions, for example, the cerebellum or the cortex. So with the help of Anu, a grad student in Yosef's lab, I used Magique to quantify LSVs with differences in splicing across the human brain, and then applied my classifier algorithm on the LSVs and splice graphs to identify what types of splicing events are alternative. And what I found was the most common modules across the brain correspond to simple binary events. On the x-axis here, I'm showing what kind of event is in the module, and on the y-axis, the count of the number of modules. So the most common module on the left are binary intron retention events. The second most common is cassette exons, followed by alternative 3' and 5' splice sites, and alternative first exons. This other bar on the right includes binary modules, for example, 224 tandem cassettes, which is what I showed earlier is what occurs in the COL11A2 gene. However, the vast majority of these other modules in the brain correspond to complex modules, meaning there's at least 3 or more isoforms in the module. And what I found during development of the classifier is that all this complexity can be distilled into simple combinations of binary-like events that overlap. For example, 1,083 of the modules correspond to cassette exons that overlap to intron retentions. And there are a lot of other interesting results that are coming out of this analysis every day. So if anyone is interested, please let me know later, and I'll be happy to talk your ear off about it. But I don't have time to go into it right now. Anyway, as I mentioned, the classifier algorithm was motivated by the challenges I ran into while classifying ESRP1 dependent splicing events. And another thing that I learned from the ESRP1 story is that knocking out splicing factors is a very useful technique for studying alternative splicing. And there happens to be a great repository of RNA binding protein knockdown experiments from the ENCODE consortium, where they have over 250 RNA-Seq experiments, and I'm showing here the gene targets that they knocked down in this word cloud. And the RNA-Seq from these experiments is widely used by researchers studying alternative splicing. However, as you scale up experiments to this size, you're almost inevitably going to run into problems like batch effects. And batch effects introduce technical noise into your experiments. And they can be caused by things like different laboratory technicians performing the experiments in different ways. Maybe one technician added a bit too much of some reagent, or another technician left the samples out at room temperature for a bit too long. And we know that these kinds of batch effects do impact splicing quantifications. This is a heat map showing Pearson correlations between all 250 RNA binding protein knockdown experiments from ENCODE. And the colors on the bottom indicate which batch the experiment came from. And as Eric van Nostrand pointed out in this paper, there's this striking positive correlation in psi within batches. There's a problem. However, there's no generalizable tools that can remove these batch effects from datasets like ENCODE. So to address this need, I helped develop a tool to model and remove batch effects from psi quantifications, which we're calling Moccasin. And this work was done in collaboration with a previous graduate student in Josef's lab, Barry Slav, and our beloved developer, Paul. And this is a high-level intuition of what Moccasin does. It solves a linear model that includes experimental covariates of interest, for example, which samples are controls versus the knockouts, as well as batch variables. For example, which samples were processed by technician A versus technician B. Then, after solving for the coefficients, it simply removes the batch variable and recomputes psi. Now, this isn't actually what's going on under the hood. For example, we are actually operating at the read count level, where the reads are scaled to psi space, and then after recomputing psi, it projects back to read count space to preserve the original coverage in the LSV. But hopefully you get the general idea. So back to ENCODE. This again is showing how ENCODE has this batch effect. This is plotting a uniform manifold approximation and projection, or UMAP analysis, which is a two-dimensional projection of the splicing data, where each point in the plot represents a sample from one of the splicing factor knockdowns, and the colors represent which batch the knockdown was performed in. The distances between points represent similarity in splicing quantifications. As you might see, the knockdown experiments are clustering together by batch colors. In fact, the square and the circle are highlighting knockdown of the same exact RNA binding protein, U2AF2, but they're more similar to other knockdowns from the same batch than they are to each other, which means the technical noise is washing out the biological variation of interest. In fact, 46% of all variation across ENCODE is due to batch effects. After I applied Moccasin to the dataset, however, the percent of variation due to batch is reduced down to 4.3%, and as you can see, the samples no longer cluster by batch, and the two U2AF2 knockdowns are now more similar to each other than they are to other knockdowns. So I hope I clearly showed why batch effects are problematic for splicing analyses and how Moccasin addresses this challenge. I also showed you how the classifier algorithm facilitates researchers to focus on the most biologically relevant isoforms in their data. And what's great about the classifier is that it automates the kind of work I did for the ESRP1 story, where I wanted to classify the types of splicing events that were disrupted in ESRP1 knockout mice. And researchers often want to do this kind of simple analysis where you're trying to find, not so simple analysis, where you're trying to find the needle in the haystack. And in this case, the needle was ESRP1 dependent splicing of FGFR2. But heterogeneous or complex data often gets in the way of this kind of analysis. So the approaches and tools I developed during my Ph.D. help overcome the challenges associated with complex and heterogeneous datasets by dissecting out the most biologically relevant signals. So today I talked about how I've applied the classifier to brain samples from GTEx. But actually, this is just the tip of the iceberg. GTEx has dozens of other tissues, which I'm listing here on the right. Don't even, you can try to read them, but I promise you it's almost every tissue you can imagine. And I think it's going to be a fun challenge for someone to apply the classifier to all of GTEx and get a better handle of how complex splicing differs across all the tissues in the human body. Furthermore, there's been recent interest in studying how splicing is disrupted in cancer. Consortiums like TCGA and Target are rich sources of RNA-Seq from a wide variety of cancers. And I think Moccasin and the classifier are going to greatly facilitate study of splicing in cancer. Actually, I don't have time to show you today, but I've also applied Moccasin to a pediatric lymphoblastoma lymphoblastic leukemia dataset that had previously unknown batch effects. And now a postdoc and I, Matthew, are helping collaborators identify potentially new immunotherapy targets from this dataset. So with that, I'd like to do some acknowledgements. So first, Kristen and Joseph, thank you so much for guiding me throughout my PhD and making me a better scientist. And thank you also for the occasional entertaining dance moves, Joseph. And don't worry, Kristen, I don't have any pictures of you dancing, but I do have lots of pictures of you doing this. And every time we saw Kristen with a bottle of champagne at lab meeting, which was pretty often actually, we knew this meant good news. But whiskey, on the other hand, whiskey at lab meeting only happened once. And that was the day after a certain president was elected. And to be honest, the hardest part of a PhD is getting through these undeniably low moments. And it's impossible to do a PhD alone. So from the bottom of my heart, seriously, thank you to everyone that helped me through this. Thank you to everyone in Joseph and Kristen's lab for being so supportive of my research in the lab and for being goofy, funny friends outside of the lab too. I really couldn't have done it without you guys. I really want to point out Matthew Thompson's epic sparkly beard here at Doobies. I also have to thank other friends and family, the Mellingers, which are like a second family to me in Lancaster. Going to their farm is always a refreshing break from graduate school. It's easy to forget about school when you're playing train game tournaments, playing music, playing with goats, and eating goat cheese. Check out their website. They deliver to Philly. And by the way, there's a goat at their farm that's named after me, which is one of my prouder accomplishments. And of course, I have to thank all my friends at Penn, in CAM and BMB and GCB, and outside of Penn in Philadelphia for the countless adventures we've been on. And I'm just going to flash through these pictures just to show how lucky I am to have all these awesome friends. Seriously, thank you for constantly reminding me that the purpose of life is to enjoy delicious food with great friends and drink way too much wine on weeknights. I also have to give shout out to my friends, Ravi and Jamie. They're my brothers. I hope we all end up in the same city again soon. Of course, I have to thank my parents for being rock solid supporters. For example, one time they drove 3 hours down to Philly to help me move in to a new apartment in the middle of my prelim exams. And yes, that's a 17-foot U-Haul truck. I somehow was able to navigate through the tiny streets of Graduate Hospital. And finally, I need to thank my two partners in crime, which are Mika and my awesome wife, Victoria. Thank you for supporting me throughout this whole process and for bringing constant dog-like enthusiasm to my life every day. I mean, jokes aside, Victoria, thank you for taking care of me every time I retreated into my PhD cave. If it weren't for you, I would have had to survive off of leftover seminar cookies and microwavable ramen. And instead, I got gourmet French meals. And also, thank you for dragging me out of the lab for adventures every weekend. You showed me that life is too short to lose track of time in the lab. Life with you has been wild and funny, amazing. I can honestly say that I'm really excited about our future. I'm really looking forward to whatever the next stop is. As long as it's with you, I know it's going to be great. And so finally, I have to acknowledge my funding sources and thank all my collaborators from over the years. And with that, I'd love to answer any of your questions. Thanks. I don't think we actually said you can ask questions in the chat.
Yes. So I will take over here first to say wonderful job, Caleb. And I will also acknowledge that Joseph and I went through some of his talk slides yesterday and told him to take out most of the detail. So while Caleb's talk did a beautiful job of representing all of the wonderful impact he's had on science, it didn't go into a lot of the nitty-gritty, but I know that Caleb would be happy to answer questions about that now or later. And so while his thesis committee gets to grill him a bit in 10 or 15 minutes, right now the chat is open for people to ask questions from the audience, and I will read them so that it makes it a bit easier for people to hear, and they feel free to chime in if I butcher your question. I also want to, while we're waiting for questions, just acknowledge family from Caleb who joined in on this. One of the nice things about doing this virtually is that we can have people from near and far. I know that there's some people even from Europe on the call, former members of the SS Lab, perhaps other friends and family. So welcome all, and thank you all for your support of Caleb throughout all of this. All right, questions. So, so one question coming in from Max is, can you talk a little bit more about how Moccasin does the correction? That is, read space versus size space, exactly what is, how the correction is working.
Yeah. Yeah, so it takes as input, I don't know if you're familiar with a combat, but kind of a similar idea. It takes as input, kind of a matrix of reads by samples, but and where each row is a junction. We group the junctions together as LSVs, and then psi is computed just like I explained earlier. And psi is then used to scale the read counts down into size space, 0 to 1. And then we run the linear regression. And then after removing batch and recomputing psi, we now have a corrected psi, which we multiply back into the original read number to give read count space again. But if you sum up the total number of reads, it's still going to have the same original coverage that the LSV had coming in. I don't know if that answers the question.
Max, way back in on the chat if that didn't answer the question. No, Max says it totally answered it for him. All right, so Peter Choi has a question asking, so first, great job, Caleb, and then asking about the classifier. What's your sense on how you set the thresholds to decomplexify? I know you've thought quite a bit about that.
Yeah, so yeah, this toy example, I said that I used absolute read numbers to decomplexify this graph, but that's not actually how the default of the algorithm works. I just did that for simplicity's sake. After lots of testing, I find that the setting that makes the most sense, like passes the eyeball check every time, is to use a threshold of 5% psi. So, for example, if we looked at the splice graph and you use 5% psi to decomplexify, these two junctions would be retained, and you'd have a more complex, bigger module. I find that if you use read numbers, then different genes have different expression levels, so you'd have to change your read number thresholds for each gene. And you can also decomplexify with delta psi, or differences in splicing, which at the surface might seem like a good idea, but what I found was there was lots of thresholding problems where a junction, for example, this junction here of including the alternative exon, would just barely pass the threshold, and this junction will just barely not pass it. And now you look like you have an alternative first exon instead of a cassette. So I find it's best to use psi to decomplexify. That was my finding.
And sort of to follow up on the classifier, can you expand a bit more on how the classifier bridges the gap between the LSV sort of raw output that you get from Majik and the way people classically think about splicing events, cassette exons, front retention, etc.
Right. Do I have a good graphic of that? Well, so for example, this module here would be represented in Majik as with two LSVs. There's an LSV that's a source on exon 7 and an LSV that's a target in exon 9. And so after we identify the modules from the splice graph, we go back and figure out, oh, in this module, there's this LSV and this LSV. And in the output of the classifier, we say this module includes this LSV and this LSV. And LSV source 7 quantifies this junction and this junction. And LSV target 9 quantifies this junction. I hope that answered the question.
Yeah, so basically, you're taking an output that isn't intuitive for those thinking about classical or traditional splicing events and interpreting it in a way.
Right.
That
Right. I was brought up in the lab environment where we thought of things in terms of these local splicing variations from the point of view of an exon, but actually classically the way splicing has been studied for decades is thinking of this event as an event, a cassette exon, and breaking it up into pieces like LSVs is not actually intuitive to the rest of the splicing field. So I was trying to bring back intuitive, interpretable outputs for the splicing field.
I will say, as someone who's from the traditional splicing field, when Josef first came with this concept of LSVs and how wonderful it was that it found so many complex things, I think my input was, that's great, but people don't want complex things. People want simple things. So you've sort of, I've watched the lab go back and forth from being able to discover everything to now trying to figure out how to give people what they want. Doug Epstein is asking, do you see any correlation between simple and complex events across tissues? And if so, might that help you in identify regulatory, RBPs that regulate these events?
Right. So I'll first say we haven't dived into the rest of GTEC's tissues yet. We've just looked at the brain. But yeah, some work that now Matthew Gazzara, a graduate student in Josef and Kristen's lab, is doing is starting to look at the output of the classifier, and we're finding some consistent differences between cerebellum and a lot of the other brain subregions. And we're finding that these consistent differences, there's enrichments around alternative exons for known splicing factors being important for the brain, like NOVA. And now, so we're pretty sure we have a good data that gives positive results that we expect. Now we're starting to dive in a little deeper and see what we can find that's not known before.
And speaking of Matt Gazzara, he has a question of what thresholds, so what's a lower limit of number of data sets that is can be inputted into Moccasin? That is, obviously it's great for huge sets like target or encode, but if someone has sequence things in 2 batches of 10, is that something that can be used?
Right. Yeah, so that's a great question. That's something I'm going to be finding out and quantifying actually in the next couple of weeks for revisions for the Moccasin paper. We've tested it on data sets, simulated data with 16 samples, and it worked very well. But obviously not every data set has 16 samples. I'm going to test it on smaller subsets, 8 or maybe 4, but that's yet to be determined.
And then do you have a sense of why you see intron retention being such an abundant, prevalent pattern? And the literature has really varied in whether people see a lot of intron retention or not.
Right. Yeah. So, one of the big things that I don't show clearly here, but a lot of the events, a lot of the modules that are complex because there's introns. And, this is a hard question because the way that MAGIC was developed and tested was actually based off of the gold standard in the field to test or to study splicing. And that's to use RT-PCR to look at, for example, cassette exons and alternative splice sites. But there's no great experimental technique to quantify introns as well as there is for intron retention, as well as there is for cassette exons. So right now, MAGIC is quantifying introns from the data, but we're still not 100% sure how, we're still, it's something we're actively working on. The field has struggled with this quantifying of introns from RNA-Seq. And yeah, so I don't know for sure if brain is going to be the only tissue that has a lot of intron retention. But it certainly seems to be a signal that's in the data.
All right, other questions? All right, if not, Caleb, wonderful job. Congratulations. Look through the chat when you get a chance. I just tried to download it for you because there's lots of congratulations from lots of friends and family. But at this point, thank you everyone for joining us. Caleb's now going to vanish into a room with his committee to be grilled a bit more, but I'm sure he will be available for texts, calls, etc. later on. So congratulations again, Caleb.